Hi, welcome to this ECIS presentation dealing with real-time electroporation and monitoring. This is one of two topics that will be dealt with in these presentations that involve the ECIS elevated field module. In addition to electroporating cells, we also can kill cells on the small electrode in a wound healing assay to monitor cell migration. But that will be the topic of another presentation. When we make the normal ECIS measurement, as shown in the upper panel here, we're applying electrical current in the microampere range. These currents result in voltage drops across the cell membrane of the order of a few millivolts. And since they're oscillating back and forth in this AC measurement, the cell is unable to detect this small change in the polarization of the cell membrane. And the measurement is essentially non-invasive. We electrically eavesdrop on the cells. But should we increase this current considerably, as shown in the lower panel, 1,000-fold higher into the milliampere range, the voltage drop across the cell membranes can be the order of a few volts. And this can result in the breakdown of the cell membrane in a well-known process called electroporation. If the current is applied for a short period of time, for example, 1/5 of a second, these pores will close up and the cells can recover. But during this pore formation, it's possible to introduce membrane impermeable compounds into the cells. Now, ECIS is not a preparative technique because it's only the cells on the small electrode that are electroporated. But it is a very interesting analytical technique, as I think will become clear as we go through the remaining slides. First, let me convince you that we can introduce membrane impermeable compounds into the cells upon the ECIS electrodes. In this slide, you see 4 cutouts of the ECIS electrode that have been exposed to this high field pulse for different periods of time, from 50 to 500 milliseconds. Here, though, we've added the membrane impermeable dye, lucifer yellow. And you can see, whereas at 50 milliseconds, very little of this dye is taken up, by 200 milliseconds of this pulse, the cells will allow the dye to enter through the pores that are formed by electroporation, and we can see this in these fluorescent images of the cells. This method isn't restricted to just small molecules like lucifer yellow, for here we see labeled dextrans at 75 and 250 kilodaltons, clearly being able to enter the cell upon, again, a 200 millisecond high field pulse. And other researchers have shown that it's possible to introduce DNA constructs by this same method. This slide from Joe Wegner's lab in Regensburg, Germany, points out that this works with a variety of cell types where we're seeing the introduction of 250 kilodalton labeled dextrans. But what is unique about ECIS is that in addition to be able to electroporate cells, it is a monitoring device. And this allows us to do an assay that I like to refer to as the CREAM assay for cell real-time electroporation and monitoring. And I'm going to show you 2 examples of membrane impermeable compounds that we introduce into the cells and then monitor their effect. These will be cyclic AMP and bleomycin, the chemotherapeutic agent. So let's first consider cyclic AMP. And first of all, I'd like to show you that the presence of cyclic AMP in the cells does affect their morphology. And we're going to show this first by looking at a signal transduction assay using the beta adrenergic receptor. Here, cells have been grown to confluence and then exposed to the agonist for this receptor, isoproterenol. And in the inset B, you can see the addition of 1 micromolar of this compound results in a striking increase in the cell impedance. Again, this is not due to the cyclic AMP directly, but the signal transduction cascade it sets up that is changing the cell morphology. To prove that this is due to the cyclic AMP that is generated in this signal transduction pathway and not some other effect of the isoproterenol, look at the trace in A. Here we begin by adding ALP, an antagonist for this receptor, followed by the isoproterenol. And notice, since we've added the agonist before, or the antagonist before the agonist, we no longer see the response that's present in B. Instead, the curve remains flat. If, on the other hand, though, we boost cyclic AMP with the introduction of forskolin, we can regenerate the response seen in B. Let me show you the cyclic AMP in another way. Here we're looking at a membrane-permeable analog of cyclic AMP, CPT cyclic AMP. And we're looking at a dose-response curve where the addition of this compound to confluent layers on ECIS electrodes results again in this striking increase in impedance. So cyclic AMP can affect cell morphology and can be deadly. Now to the key experiment in this series. Here we're going to use 8-hydroxy cyclic AMP. This is a membrane impermeable analog of the compound. There are two traces here. First, the blue trace, which is not going to receive the cyclic AMP, but just buffer, and the red trace that receives the membrane impermeable cyclic AMP compound. So those are added, either buffer or buffer containing the compound, at arrow 1. Notice there's a slight response of the cell due to probably temperature changes and shear stress, but it quickly comes back to control levels. Then at arrow 2, we apply an electroporation pulse to both wells, those containing just the buffer and medium, or those containing the buffer and the cyclic AMP compound along with medium. Notice that in the case of the control, the blue trace, the electroporation is quite an insult to the cells, and there is a morphological response as these curves drop down but quickly come back to control levels in about half an hour. Look at the different response, though, in the red trace. Again, the curves begin this downward trend upon electroporation, but upon the entry of the 8-hydroxycyclic AMP, they go through this increase in impedance that we saw in all of the previous slides. Again, this is cell real-time electroporation followed by monitoring. In the last experimental example here, we're using bleomycin. This is a membrane-impermeable chemotherapeutic agent. Here, the black trace does not contain bleomycin, but has been electroporated shortly before 2 hours into this run. Again, as we saw in the previous slide, electroporation results in a drop in the impedance that quickly comes back to control levels, again, in about 30 minutes. The red trace has bleomycin present in the medium, but is not electroporated. But the blue and green traces, with bleomycin present in the medium, are electroporated, and notice that these curves drop down, but then stay down at low impedance values. In fact, it's not until about a day later that these curves return to control levels. Well, that's what we can do with the ECIS cream assay. You may consider it for the introduction of nucleic acids, for example, small RNA molecules, and perhaps look at RNA interference with the system. It is a unique opportunity to combine these two events in one simple assay. Our website has more information about this type of assay. You can go to biophysics.com, look at publications, and then circle electroporation. Well, I'd like to thank you for joining us for this presentation. And on this slide, you can see the names of people and email addresses you can contact. If you'd like more information about ECIS, perhaps you'd like to demo an instrument or need information about pricing, we'd be happy to help you out. So thank you for listening.